I'm feel honored to introduce Dr. Prince Sharma from Punjab University, Chandigarh. Is a President Association of Microbiologists of India, former Dean Faculty of Sciences, Punjab University, and Fellow Academy of Microbiological Sciences, India. So he has a 37 years of teaching experience, developed several new syllabus and pedagogies. So diverse research area like reverse vaccinology, antimicrobial resistance, engineering and marketing of industrial enzymes, and biosensors for microbial diagnostics. So he has published 132 research papers, and H-index is a 42. So, and completed 14 research projects with the funding agencies like a DBT, DST, UGC, ICMR. So, and guided 29 PhD students, and right now he is guiding 8 PhD students. He received many prestigious awards like a GlaxoSmithKline Vaccine Award for work on our reverse vaccinology. So research paper on reverse vaccinology in a scientific reports was selected in the top 100 papers during the 2017. So received Prem Lata and Professor DVS Jain Research Foundation's Best Research Publication Award during 2020 and 21. And he is also recognized as a distinguished scientist, as is in the list of 2% scientists. So in the world, in the years of 2022, 2025, and recognized as by the Stanford University. So we welcome Dr. and Professor Prince Sharma. Okay, thank you.
So, good afternoon, respected panel members, everyone. Myself Mustafa Vohra, and I am going to today present you my PhD topic, a multi-omni approach for understanding biofilm formation and biofilm-associated antimicrobial tolerance in Pseudomonas aeruginosa. Now, this is the presentation outline. I will start with introduction, and I will present you my results and discussion based on different objectives. I will conclude and then present my limitations of the study. So, starting with introduction, Pseudomonas aeruginosa is an opportunistic pathogen. It causes several nosocomial infections, especially in immunocompromised patients. It is also one of the WHO critical priority pathogen, and it has a high antimicrobial resistance rates. The reason is because it forms a structural microbial community known as biofilms with the help of extra polymeric substance, and these biofilms accounts for more than 80% of chronic and device associated infection. There is increase in almost 10 to 1,000 times more resistance in biofilm as compared to planktonic cells, and this is because the biofilm provides a physical barrier. It also results in metabolic heterogeneity within the biofilm, and it also increases the enhanced genes transfer. Now, these are the basic stages of biofilm formation. It starts when planktonic cell finds the surface and adheres to the surface, and the motility is downregulated, which is called as initial attachment phase. After a certain while, when the bacteria has has decreased its motility, it proliferates and attaches to each other to form microcolony. Then after that, as the microcolony grows, it forms a 3D structure consisting of heterogeneous population, which are called mature biofilm, and finally cell are released from the biofilm and they revert back to planktonic lifestyle, and these are called this is called as a dispersal phase. Now, why we selected study? So we found that during our literature we found high variability in the reported biofilm phenotype and gene expression across different study, and there was very limited evidence linking morphogenical changes during biofilm development with phase specific gene expression patterns. Most of the study were done on Pseudomonas aeruginosa PA1, which was a standard strain. Lack of standardization also we found, including difference in growth media, surface material, and major limitation we found was the poorly defined endpoints. Most of the study restricted their studies at mature biofilms, basically around 12 to 24 hours, but we found that phase specific genes were overlooked during this mature biofilm sampling points. And finally, we didn't found any robust model that systematically integrated biofilm development with antimicrobial resistance. Now, why we selected multi-omics? Because most of the study were done using single omic approach, and single omic gives an incomplete biological insight due to only focuses on one molecular level. Biofilm as such represents a complex multi-layered adaptation, and hence there was need to capture the molecular dynamic at all the level, DNA, RNA, protein and metabolite, which multi-omic integration does, and it also provides a comprehensive perspective on different biofilm stages. So with that understanding to understand the biofilm at system level, we did genomics, which identified strain specific genetic element, transcriptomics, which reveals the gene expression, proteomic captures the translation and post-translation modification, and metabolomic identifies the final functional output of the cellular processes, and SEM microscopy visualize the spatial architecture. So with that approach, we used multi-omic approach to study the biofilm formation in Pseudomonas aeruginosa. So based on this, our study was divided into 4 objectives. First phase was to screen the clinical isolate, evaluate its biofilm forming capability, antimicrobial resistance, virulence, etc. Second phase was to optimize the biofilm methodology. Third was to conduct a multi-omic analysis, and the fourth phase was to do a comparative meta-analysis where we integrated experimental finding with systematic literature review to create a comprehensive biofilm developmental model. So we'll start with first objective. This was the methodology used during the first objective. First of all, clinical were isolated from the hospital, around 84 Pseudomonas aeruginosa clinical isolated from patient specimen identified using Vitek system, and standard strain PA01 was used along with the clinical strain, and working culture of 2.5 into 10⁴ was used throughout the study. Antimicrobial susceptibility testing was done using Vitek system and diffusion. Then detection of presence of gene was done using RT-PCR. Biofilm genes were detected, virulence and resistant genes were detected. Biofilm formation of these isolates were checked using crystal violet method. Statistical analysis was carried out. This study was also approved by the Institute Ethical Committee. Now these are the few results of objective 1. First of all, antimicrobial resistance within the 84 clinical isolates. We find that majority of the isolates were quite resistant to the 4th generation of cephalosporin, especially ceftazidime, cefepime, and also they were resistant to ciprofloxacin as well as piperacillin tazobactam. Then we also checked the gene prevalence, and we found that most of the isolated carried biofilm related gene such as ALGR, APRA as well as virulence related gene. Resistant genes were varied among the strains, with beta-lactamase gene almost found around 30% of the isolates, and efflux genes were found around 50 to 60% of the isolates. This was the crystal violet assay used for screening of biofilm formation, where we prepare a culture media, dispense into microplate, then we incubate the bacterial culture to form the biofilm. After that, we rinse the planktonic cell and then we stain it with crystal violet. After staining, we again wash and dry, de-stain it with 70% ethanol, and we read the absorbance to get a quantitative analysis of how much biofilm formation is formed. So with this method, we found the biofilm production capacity among different isolate. We found that there were 4 categories of isolate based on the absorbance, with strong producers in almost 14%. Most of the biofilm producers were moderate and weak producers. There were just 4 strains which we found to not produce biofilm formation. Then we come to second objective, that is methodological optimization. Here we divided our experiment into 5 different phases. First one was to assess different phenotypic method for biofilm formation. Second was to understand surface interaction, where we used 26 isolate, 4 different surfaces. Third one was to study the surface dependent gene expression, where we used 4 biofilm forming gene and 3 stains and 6 different surfaces. Fourth one was to check the biofilm removal method, and fifth one was to time resolve phenotypic and transcriptomic profiling. So this is the result of the first method, where we use crystal violet as well as rhesus urine and Congo red method, which are commonly used for phenotypic biofilm detection. We found that all 3 methods gave a different kind of resolution within the biofilm. Crystal violet gave majority of biofilm producers, rhesus urine was giving less amount of biofilm producers, while Congo red was only giving whether it formed matrix or not. But finally we came to realize that crystal violet detects biomass, rhesus urine detects metabolic activity, while Congo red detects matrix. So all 3 which are compared in the literature, we can't compare here, and for all 3 different, we can use all 3 methods for different purposes. Then we also used a Calgary device for studying a biofilm formation. So this is general protocol, where we used lead. Here we specifically use sterile PCR plates as a lead in a deep well plates. We grew the biofilm in deep well plates and covered with the biofilm lead, and then we estimated how much biofilm is formed with the help of crystal violet. So this was the second experiment, where we checked biofilm formation in different surfaces. Tube was used, Calgary device was used, where we used PCR plate lead to form the biofilm formation. Microtiter plate was used, as well as catheter was used. And this was our result of what was the biofilm strength across different surfaces. So initially we just checked using microtiter plate and 3x replication, but we increased the replication number of replication to 6x. We find that there is a difference in the strength. So the strength we measure using crystal violet depends on what surface is used, how much replicates you have kept, because it all depends on how much washing you are giving. If we give high washing, then biofilm are easily removed, especially in microtiter plate. And thus we found that within the Calgary device, almost the no biofilm which we found in microtiter plate was almost not found, and all the strain were forming biofilm production. This was the variability among clinical isolate. Based on different surfaces, we found strain to produce different biofilm strength as per the crystal violet absorbance. But there were a few strains which were giving quite similar biofilm strength across the surfaces, such as PS51, which was giving weak strength across the different biofilm surfaces. PS120 was giving strong biofilm strength across the surfaces, and standard strain which was giving a moderate biofilm strength across the surfaces. So we selected these 3 strains for further studies, where we wanted them to form a good equal biofilm across the different surfaces, so that we don't find variability where we study the omic methods. Then we studied the gene expression, surface dependent gene expression among these 5 different surfaces: steel, different types of plastic, glass, as well as latex and PVC catheter, to understand how these different surfaces leads to difference in gene expression. So these were the results among 4 genes, PAL-A, ALGR, PSL and LAC-A, and we were testing 3 strains across 6 different surfaces. We found that especially in latex and PVC catheter, the expression is actually quite high as compared to glass and steel. And when we did statistical analysis, we found that surface type were the major contributor in variance, accounting for 42% of variance among the biofilm among the surfaces. Then our 4th experiment was to test biofilm removal technique, where we use different biofilm removal method. We wanted to check which method gave us the highest viable cell with minimal cell damage. Since we were going to perform omics analysis later on, we didn't wanted cell lysis to occur while we are removing the biofilm. So for that we tested scrapping, swabbing, sonication and combination of scrapping and sonication. We found that mechanical scrapping gave us the highest viable cell recovery with minimal cell disruption. So this was the question we asked during experiment 1 to 4. Do different phenotypic method estimate the biofilm equally? We found that combinations of assay are required to overall study the biofilm with the help of phenotypic assay. Does biofilm strength vary across the surfaces? We found that surface significantly affects the biofilm distribution. How much do surface influence the biofilm gene expression? So we found that surface is the strongest driver of gene expression. And does the biofilm recovery method impact the cell lysis and omic analysis? We found it does, and scrapping yields the maximum viable cell with least cell damages. Now this was the final method of our checking of various biofilm method, where we wanted to understand at what time interval should we collect the samples. So for that we conducted a time resolved phenotypic and transcriptomic profiling, where we used 3 strains grown in different grown and planktonic as well as biofilms with and without 1/4 MIC of ciprofloxacin, and we collected sample at different time interval, and within that time interval we did crystal violet staining, metabolic rate as well as crystal violet quantification, and based on that preliminary analysis at 4 different developmental phase we performed qPCR expression and antimicrobial resistance assay. These were the 3 strains we had selected for a further study. PS120, which was a strong biofilm producer, which was a multi-drug resistance, had the higher MIC of greater than 4 microgram, and it was a clinical sample. PS120 was weak biofilm producer, susceptible, and was resistant to ciprofloxacin and was isolated from urine, and this was the standard strain which produced a moderate biofilm production. So this was the strong biofilm producer. As you can see, it forms a huge amount of matrix as biofilm formation, as biofilm is forming within the 24 hours. This is PS21. It forms a little bit less biofilm, quite less as compared to the one produced by PS120. And this is biofilm formation with the standard strain, which also resulted in good amount of biofilm matrix in the supernatant. So in this experiment, to understand how biofilm is progressing at different time interval, we used a PCR strip tube. As I was working in RT-PCR lab, I understood that since for Calgary device we are using PCR plate, we can use different strips, and we can remove the strips at different time interval in which biofilms are grown, and we can do staining, and based on that staining we can understand how biofilm is progressing at different time interval. So this assay revealed that 3 strains produce different amount of biofilm as the time progresses. PS120 with ciprofloxacin and untreated started forming biofilm within 2 hours because it was ciprofloxacin resistance, and it gave a high amount of biofilm around 22 to 24 hours. Now PaO1 and PS51 both showed a delay in biofilm formation in ciprofloxacin, and around 16 to 12 hours it started forming biofilm, and around 26 hours it reached the biofilm as compared to untreated one. And visually we found around 56 hours were taken to slowly see the dispersion phase among this biofilm forming strain. So these were the results of crystal violet which we checked for biomass production as well as metabolic activity, which we checked using resazurin, and we found that PS120 formed a high biofilm, then PaO1 and then PS51, though at the later stage it almost formed the same amount of biofilm as the time progressed, and same goes with metabolic activity. Initially all three strains were quite differentiated with the metabolic rate, but at the end after 40 hours it almost kind of showing same amount of metabolic rate. Now we did gene expression analysis at the 4 different time interval where we saw attachment, microcolony formation, maturation as well as dispersion. So when we visually saw when attachment was seen, when we can see microcolonies, when we can see 3D structures, and when we could see dispersion, at that time interval we selected the sample and we perform a gene expression analysis of 29 key genes and 3 strains, 4 different time intervals with and without ciprofloxacin. Now the aim of the gene expression study was to study gene expression was normalized to attachment phase as a reference point when we were studying different phases. For studying comparing strain gene expression, we normalize the gene expression to PaO1. For studying antibiotic exposure, we used a reference test as a normalized reference point. And these were the different genes related to matrix, regulatory and resistance, which we checked for gene expression analysis, and 4 different interval. And the question we asked was, how does gene expression changes across the biofilm phases? How does ciprofloxacin exposure modify the expression? And what are the strain specific transcriptional signatures? So quite comprehensive results we got. This is just few of the results which I am showing. So this is the phase dependent gene expression of PS21 during the different phases. So as we can see, as compared to attachment phases, the regulation of matrix as well as motility genes are quite high. The regulatory genes are differently expressing at different phases, and as we can see, as the biofilm is forming at different phases, biofilm dispersal genes are also, expression of dispersal genes are also increasing as compared to attachment phase. Then we compared PS51 and PS120 gene expression with relative to PaO1, and we regularly found that PS120 was showing higher gene expression, which was a high biofilm producer, as compared to PS51 across various regulatory, various category, including biofilm matrix, motility, as well as around iron and nitrogen regulation and quorum sensing. Then this was the results of how ciprofloxacin affect the gene expression. Here we can found that this PS51 had almost low expression during the microcolony phase, and as it increased, as the biofilm progressed through maturation and dispersion phase, then the expression increased. While in PaO1 and PS120 we found that usually the gene expression with related to different genes in presence of ciprofloxacin are quite high during the attachment and microcolony phase, and it decreases as the biofilm progress through maturation and dispersion phase. Then we integrated the phenotypic results we obtained with the transcriptomical response, and we found that PS120 shows minimal changes, high biomass and high metabolic activity, and it shows early suppression and late activation in maturation phases. PS120 shows a delayed initiation in presence of ciprofloxacin, delayed metabolic activity. During the maturation phase, these are the specific genes related to matrix and antibiotic resistance which are high. And PS51 shows a delayed but paradoxical enhanced biomass at 20 to 36 hours. So here we see that in ciprofloxacin the biofilm is high as compared to untreated strain, especially during 22 to 36 hours. Though ciprofloxacin slowed the initial attachment phase. Then we come to objective third, which was our multi-omics study, where we wanted to understand how these changes in gene expression as well as transcriptomic, proteomic, metabolomic phase occurs at different biofilm phases. So this was the overall concept, where we selected standard strain and clinical isolate, grew at different phase, and we collected sample and we performed electron microscopy, transcriptomic, RNA sequencing, proteomic and metabolomic. So first we start with genomics. We did the whole genome sequencing of the 3 isolate. So we extracted the DNA and we perform library preparation and sequence using the Minion MK1C, and we then did different types of bioinformatic analysis with respect to these 3 different biofilm forming strain. We did assembly, we did Busco analysis, then we did taxonomic profiling using Krona, phylogenetic analysis. We also studied the structural variation analysis. Then we wanted to study is there prophage present within the strains or not, and finally we perform functional annotation. So these are few of the results of the bioinformatic analysis of the 3 strain, and as we can see that PA1, which is the standard strain, shows almost 99.98% with the reference strain, while PS51 shows 96% similarity and PS120 shows 95% similarity. PS120 has a high amount of gene as compared to standard strain, and it has number of contigs are high as compared to PA01. Then this is the genomic feature crisscross plot, where we find that the outer one, which is the standard strain, red is the similarity between the 3 strains and green is the variation. So we can see certain variation in PS51, but we find a large number of variation found within PS120 strain as compared to PA01 strain. Then we did phylogenetic tree analysis, and PA01 was found in the standard strain clade. PS51 was found in the AZPA clade. This is the standard strain. This is the PS51, which is found around AZPAE clade, which is an hospital adapted lineage. And PS120 was a multi-drug resistant strain which belonged to nearby of PASG NDM clade, which are all highly antimicrobial resistance pathogens, and these are quite globally distributed strains. Then we did prophage visualization. Here we can see that PA01 only contains 3 different prophage regions, while I guess PS120 has around 11 different prophage regions, and PS51 has also high amount of prophage regions as compared to PA01. Then we studied the specialty gene among these 3 strains, AMR, virulence and biofilm. Here we can see that AMR genes as with related to efflux pumps were quite similar within the 3 strain, but aminoglycoside enzymes were present high in PS120, which was a resistant strain, and also general resistant genes were high, and betamet resistant genes was high among PS120 as compared to other strain. Now when we see biofilm related genes, we find that PS120 was kind of low in when we see related to adherence, exopolysaccharide and flagellar motility, but quorum sensing genes were present high in PS120 as compared to other strain, and virulence genes was also found among the 3 strains. Then we performed genomic variation. across biofilm, different biofilm motility, toxin secretion, energy and regulatory pathway. So here we can see in detail that where all different genes are present. Here we can see biofilm signaling and quorum sensing genes are quite high when we compared PS51 and PS120 with respect to PaO1, and metabolism related genes are also quite high and virulence related genes are also quite high in PS120 as compared to PS51. Then we wanted to see mutations in biofilm associated pathway within these two strains as compared to PaO1. So we found that almost 347 high impact variants were found in PS51 and PS120 when we compare it to PaO1. Among that, PS51 had a 130 variant and PS120 had a 149 variant. Important variant related to attachment phase was seen in type 4 pili system, where PS1 disrupted had a frameshift mutation in PILF genes, while PS120 had an extensive disruption in pili, type 4 pili genes, and frameshift mutation in PLY1 and PLI genes and deletion in PLIC gene. So these were one of the important mutations we found among the strains with respect to PaO1. Flagellar system was also, we found variation among the flagellar systems where FLIQ and FLIP shared mutation was found, and additional mutations related to adhesions were also seen in these two strains. Then in microcolony and matrix formation, we find that PS120 had a PALA insertion disrupting glycosyl hydrolyze function. So there was a mutation in PALA gene and there was a shared mutation in PALI and PALF gene. Alginate biosynthesis genes also we found some mutation, and we found some alternate pathway in PS120 and PS51 which were present in this strain but absent in PaO1. And then we found also some mutations with related to dispersal phase, where we found that diglucuronide cyclase gene mutation and PHOQ sensor kinase mutation, and the stress forming genes RPO and sigma factor mutation was found in PS120. So this is a comparative analysis among the genomic variant, where we find that reference strain which we used to compare contains the type 4 complete type 4 pili functional providing synthesis and intact exopolysaccharide pathway, while PS120 shows an extensive disruption in type 4 pili, PAL pathway alteration, alternate flagellar system, and then PS51 showed a limited pili disruption, and there was a mutation affecting LPS structure, and there was a mutation affecting regulatory control within the PS51 isolates. This is the multi-omic analysis we done using the three strains, where we took PS120, PaO and PS51. We grew PS120 in planktonic phase and took sample at 12 hours. We grew PS120 in biofilm and took sample at 4 different time interval, which we obtained during the objective 2 time phase. Then PaO1 as a standard strain was grown as a biofilm. Sample was taken at 24 hours. PS51 was grown with and without ciprofloxacin 1/4 MIC, and sampling were done at 24 hours. So total 8 samples were collected of these 3 strains at different condition, and for each sampling point we did first set, we kept 2 sets. First we did SAM microscopy, and in second set which we collected the biofilm, we performed transcriptomic, proteomic and metabolomic. And these were the comparison we studied: difference between the biofilm phases. Here in PS120, difference between biofilm and planktonic phase between PS120, difference between strain at 24 hours, we compared 3 strains, and finally difference between with and without ciprofloxacin, which we studied for PS51, which was a ciprofloxacin sensitive strain. And this was the sampling collection method. So there was lots of combination studied. I'm going to give just few important results which we found during the transcriptomic and as well as proteomic analysis. So transcriptomic was done using, we prepared a standard library and performed sequencing using Illumina, and then we did data analysis. So this is a key transcriptomic result in PS120 in maturation phase genes and pathway. So in PS120 during the maturation phase we found alternate carbon metabolism, where we found gene were related to malonate processing were highly expressed, gene related to alcohol metabolism were also upregulated, and genes related to branch amino acid degradations were also regulated, which suggests alternative carbon metabolism going on in PS120. Motility matrix related genes were also seen regulated, especially ALGD, alginate gene was highly expressed in PS120. Quorum sensing gene was also finely regulated during the matrix formation, and we found type 4 pili was negatively regulated during the maturation phase, because now the biofilm is mature phase and doesn't require pili for its function. Then we also found gene expression related to oxygen limitation, where we found gene related to anaerobic respiration were highly activated, gene related to arginine deaminase, and gene related to acetyl CoA synthesis were highly expressed during the PS120 maturation phase. And we also found gene related to cell signaling found expressed during the maturation phase of PS120. Now during the dispersion phase we see also quite good metabolic adaptation. We find that during dispersion phase also alcohol metabolism is strongly regulated, as well as branch amino acid, acetyl utilization, and methyl malonate processing. These were highly regulated alternative energy pathway during the dispersion phase, and these were the top biofilm regulatory gene. PQS, LAS-R and GAC-A were regulated highly, while the ORF and lactinase gene, which is useful for surface attachment, was negatively regulated, as well as ORF gene, which is responsible for membrane modulation, was also negatively regulated. Then when we compared PS51 antimicrobial gene response as compared to control, we find that when the PS51 is induced with ciprofloxacin, 1/4 amount of MIC, we find a good amount of gene regulation related to cell wall modulation. So when ciprofloxacin is going on, there is a regulation in cell wall modulation, which is helping the PS51 grow during the ciprofloxacin. Then repair mechanisms are going on, and as well as prophage activation is seen during the PS51 ciprofloxacin related gene expression. So we find significant adaptation related to cell wall modeling, RNA repair, protein quality management, mobile genetic activation, as well as certain metabolic activation was seen when ciprofloxacin was used in presence of PS51. And when we compare these three strains, PS120 and PS51, in compared to PaO1, we find that PS120 has an extensive changes, while PS51 has a fewer changes as compared to PaO1. Most down regulated gene was quorum sensing gene was quite deregulated in PS120, and there was also reduced aromatic compound metabolism. Enhanced alternate energy were activated in PS120 as compared to PaO1, while in PS51 we see a regulatory and transport gene preferentially induced, as well as biofilm tolerance without broader gene activation. So these were the key adaptation of PS120 and PS51 as compared to PaO1. Then we also did proteomic analysis, where we collected the sample, did protein extraction, did LC-MS, and then data processing and statistical analysis were done. These were the few results which I am showing. In PS120 key maturation phase, proteomic changes, we find that proteins related to antimicrobial resistance was downregulated. Metabolism was also downregulated in PS120 mature phase. Quorum sensing was highly regulated, and stress response were, proteins related to stress response were highly regulated in PS120 mature phase. Now when we compare ciprofloxacin response of PS51, here we find that major porins were upregulated, ORF, ORPD and PORB. So porins and membrane modulation proteins were upregulated during the ciprofloxacin response, and ATP synthase were kind of deregulated, and kind of genes related to protein modulations were also downregulated, such as GROK-EL, CLPX and HCP. These are all related, all proteins related to changing the protein, this act as a chaperone. So these are also quite downregulated in presence of ciprofloxacin in PS51. These are the strain specific protein expression when we compare PS120 with PA1 and PS51 with PA1. We find that as in transcriptomic also, proteomic also, we find an higher proteomic expression of methylmalonate pathway, which we are quite regularly finding in PS120, and also related to MAX-A and iron related regulation was seen upregulated, while in PS51 we see succinate related genes were quite succinate related proteins were quite upregulated, and catalase and related genes were quite downregulated. So overall we found that PS120 is an active adapter. It reprograms metabolism, transports and regulation to thrive in the biofilm environment, while in PS51 we found it's a survivor. Usually it minimizes energy use, it's suppressive oxidative and metabolic activity, and it survives through dormancy like behavior. So these were the quite two majorly difference in proteomic expression when we compared PS120 and PS51. Metabolomics was also done. We didn't found any as such signals which were highly activated, so we didn't consider that in our final results. Scanning electron microscopy was done. These were the 8 SAM microscopy images of the 8 different samples we had taken. This is PS120 at 2 hours, hardly any biofilm is formed. PS120 12 hours we start to see clumping biofilm formation. Now during maturation phase we see less cells because the layer is highly covered in matrix. So because of that matrix, usually we are not able to see biofilm cells are there in the behind, but majorly the SAM microscopy was covered with matrix formation because it was forming high amount of matrix. Then at PS120 we see little bit slowly dispersion phase is going on, and this is the SAM microscopy when we grew at the planktonic phase. PaO1 at 24 hours, and this is PS51 grown at 24 hours without ciprofloxacin, and here we see some granulation and some shrinkage when we grow at ciprofloxacin. But overall, as we see in our objective 2, the biofilm forming equals and maybe highs when ciprofloxacin is given in PS51. And final objective was to do a meta-analysis where we wanted to integrate the results obtained in the literature with us to form a biofilm model. So we selected a PRISMA workflow where we identified 778 papers. Out of that, duplicates and other records were removed, and finally 509 were selected. Out of that, 380 were fully accessed paper and eligible for the study, and finally 152 papers were included in the meta-analysis. These were the selection criteria on basis of which we selected the final 152 paper. Majorly was what organism and what omics technology, did they use any omic technology or not, whether they compared biofilm and planktonic phase, which biofilm strain did they use antibiotic or not. So based on all these criteria, finally 152 high quality studies were selected and data were extracted. The extraction data was done comprehensive, where we extracted the metadata, biofilm biology, design, and as well as various omics approach and various functional genes, proteins, metabolite found within this paper. and we standardized the gene when we compared when we mapped it to Pseudomonas database. Proteins were mapped to UniProt database, and metabolites were mapped to KEGG compounds, and we standardized our data and the data found in the literature. These are just few examples of data we found within the meta-analysis, where we see the strain usage pattern, where we found that PA1 plus mutants were highly used within the BIOFILM-OMICS study, whereas PS14 was quite less used. Clinical strain only were also quite lessly studied during the BIOFILM-OMICS. And these were the growth media we found from this 152 paper, where most of the study was done using LB media, which we had also used for our study. Then we did model development, where we selected, now different paper at different time interval, so we selected the range. Within this time range, if the omic analysis has done study 0 to 2 hours, we selected as an attachment phase, 2 to 12 hours microcolony, 12 to 24 maturation, and any study done after 24 hours was used as a dispersion phase. And we used various visualization method and statistical method to form a model for biofilm formation. So these were the data from the meta-analysis, and our data included, where we found that with respect to ciprofloxacin, DNA damage induces SOS response, enhanced biofilm formation is seen under the stress. There is a metabolic reprogramming during the ciprofloxacin, and there is suppressed mortality and virulence when we use ciprofloxacin. And these were the majorly expressed pathways during the ciprofloxacin, includes DNA repair matrix, motility, virulence, and quantum sensing were down-regulated pathway. Then we extracted the details related to biofilm matrix and structure component, where we studied key polysaccharides alginate, PAL, and PSL. These three polysaccharides are responsible for the major exopolysaccharide production in Pseudomonas biofilm formation. And we studied the key regulatory and signaling pathway, CDGMP signaling pathway, CAMP-VIR pathway, quorum sensing pathway, and GET RSM pathway. These four pathways were studied, and matrix was studied. These are the different genes we took into consideration. This is the literature where different genes are involved in biofilm formation with respect to CMP pathway, GET RSM pathway, quorum sensing pathway, and CDGMP regulation pathway. So all these genes were extracted from our study from the literature, and then we formed a comprehensive model of how biofilm is forming in different stages. So let's start with attachment phase. In attachment phase, usually flagellar activity are actively expressed to reach the surfaces. Then usually pili A, type 4 pili is also used for twitching mortality. And then when it encounters the surfaces, there is an upregulation of CAMP and VFR pathway. Then there is a reversible attachment, and finally there is a transition state where RSM pathway is activated, CDGMP pathway is downregulated, and matrix usually is not regulated during the attachment phase. Then during the microcolony formation, surface sensing PIL-Y1 gene is activated. There is an increase in CDGMP, which results in high amount of biofilm formation. There is a regulatory switch where GET-A is highly expressed and RSM-A pathway is downregulated. There is also increase in quorum sensing pathway. Along that, there is an increase in matrix formation, and there is a decrease in mortality, and that results in cell aggregation, that is microcolony formation. These are the various key genes we found during expressed in the microcolony formation phase. Then during the maturation phase, usually signaling network usually goes on, where high GMP results in high GET and RSM is repressed, quorum sensing network is regulated, and here we also found that anaerobic pathway is also activated during the maturation phase, and denitrification also occurs. Then we found ALGD, which we found for PS120 was highly regulated, which results in alginate, and PSL pale appearance results in 3D matrix structure, and stress response is also activated during the biofilm maturation phase. Ultimately, biofilm dispersion phase, where and various environmental cues such as nutrient limitation, nitric oxide accumulation, denitrification byproducts, this results in phosphodiesterase activation, which results in downregulation of CDGMP, which results in planktonic status. Here DIGMP decreases. There is a sharp reduction in intracellular and also RSMA is reactivated. Flagellar genes are highly reactivated. We found these genes to be highly reactivated during the dispersion phase, and matrix degradation genes are upregulated. Here the cycling is complete, and again the phase of attachment microcolony maturation starts. So this is overall conclusion from the study, that clinical Pseudomonas shows strong strain specific variability in biofilm formation, antimicrobial resistance and virulence, directly affecting the clinical outcome. Surface on biofilm is grown does affect our gene regulation. Then biofilm follows a tightly regulated state specific behavior. Metabolic reprogramming and regulatory adaptation are the central to biofilm for persistence. So we usually study ciprofloxacin or any antimicrobial resistance, we study efflux genes, but metabolic reprogramming is also going on when the strain is adapting to ciprofloxacin, and sub-inhibitory antibiotic exposure leads to a high biofilm response, as we have seen in PS51 also, reinforcing tolerance mechanism. Integrated phenotypic and multi-omic analysis reveals a complex network that cannot be resolved using a single network, and system level multi-omics is essential for understanding biofilm associated infection. These are the clinical impact. First of all, in diagnostic we can use 3 different methods to study biofilm, and especially we found that biofilm can be easily detected within 5 hours. Most of the clinical studies still studied planktonic, but we found that almost 95% of clinical isolate we obtained in number hospitals were biofilm producers. We need to study as a biofilm also this isolate. Then we also can target various genes we found during our biofilm formation, especially anaerobic metabolic inhibitors, which are not quite studied, can be used to inhibit such a biofilm forming strain, and multi-omics framework can be easily used to study this biofilm formation. And finally, limitation, there were quite few limitations of this study. First one was that comprehensive multi-omic bioinformatics was not performed, as we had selected 8 different strains across 4 different 3 strains, 4 different biofilm phases with and without ciprofloxacin. The bioinformatics was quite high, so comprehensive multi-omic bioinformatics was not done. We scrapped the biofilm and we studied average biofilm expression, but as we know, biofilm are growing in layers, so spatial expression was not studied. Triplicates were not done because of the cost for the omic assay. The major limitation I found is that the closed system which we are studying, there is a limit in how we actually represent dispersion, because in dispersion biofilm cell needs to go a new place to again form a biofilm, but in a closed network where will the cells go? So dispersion dynamics are actually quite limiting when we use closed biofilm system. Multi-omics method were done on a plastic surface and not clinical surface. And finally, we used an in vitro biofilm model, and host in vivo models are not clearly represented within these results. So these are the references. 2 paper presentations were done, and 1 paper is published, and 1 review is published, where we studied comprehensive genomic, transcriptomic, proteomic and metabolomic, and 2 papers are under communication. Finally, I would like to acknowledge Dr. Sandeep Sharma and Dr. Narayan Kamat, my guide, which guided me throughout the study. They were available whenever I needed. The institution, Lovely Professional University and Sri Vinobhai Hospital. The proteomics was done at IIT Bombay, so I would like to acknowledge them. and my friends and family for their unwavering support. Thank you. Thanks.
Okay. Great job. What should I call you? Vohra or Mustafa?
Mustafa is fine, sir.
We should call you.
Mustafa is fine, sir.
Okay, Mustafa, that was great actually. A very comprehensive study. Now, before I really go further into other discussion for which I have come actually.
Sure, sir.
I wanted actually in the start only that why don't you introduce yourself first? Tell us more about yourself.
Okay.
About your education, about your place where you are actually.
Okay.
And where will you go from here now?
Okay. So I am Mustafa Vohra. I had done my BSc in microbiology from D.Y. Patil, Pune, under Pune University, MSc from Modern College, Pune University in 2009. From 2009, I have been working in various research institutes. Initially, I was involved in studying applied microbiology, ethanol formation, butanol formation from lignocellulose waste, studying anaerobic microbiology, methane production. Around 2017, I shifted to my hometown, Silvassa, which is in Dadra and Nagar Haveli. I shifted in 2014 and from there in 2017, I joined Namo Hospital, where I have been working as a microbiologist. During the COVID-19, I was the lead microbiologist handling the COVID-19 lab. And during that time, we got some sequencing PCR machine, and I got an opportunity to perform my PhD. So from around 2020, I have been registered and doing PhD. Till now, I am still working as a microbiologist at Namo Hospital, which is currently in Silvassa.
Okay, great. That's really great. Can you come to the first slide, which is your title?
Yes, sir.
Okay, so let me first tell you that you have done quite a comprehensive work on where you have done a multi-omics approach. People do one-dimensional omics approach, but you have tried to integrate all those, and that too with the clinical strains, because mostly people go for PO1 and we have done a lot of work on Pseudomonas quorum sensing, and we are working on vaccines against Pseudomonas also. So we work on AMR or of Pseudomonas also. Okay. So people just go for PAO1, where I always feel that unless you do the work on clinical strains, it's no point working on that. That part is good that you have taken the clinical strains also 84, and then you have selected out of those thing. And now when I looked at this title, you have focused on the biofilm approach. I mean, to target biofilm actually.
Yes, sir.
Your whole work is actually encompassing understanding the biofilm formation, which multi-omics approach, how it is formed, the differences in the clinical and the standard strains and all that. So it's a very comprehensively you have studied that which genes go out, metabolic way, how metabolism changes, how the transcriptome gene expression changes. Okay, that is very, very fine what you have done. One norm for a microbiologist is that when you write the name of the organism, you don't write it like that. So you always write it in the lower case actually.
But in title only, thesis title, I had all caps lock, so I kept it like.
No, that doesn't. Who stops you writing in the lower case? That's it.
At that time I didn't took into consideration. Later I wanted to change. But since in my synopsis I had done this mistake.
But now you could have done in your presentation. I had mentioned probably, but because it's a norm for the microbiologist, even an MSc student in our department, when he does this same mistake, actually, we are not happy.
Yes, sir. Yes, I understand.
Okay, that's one thing. That's one thing you should be doing. Wherever you do the next presentation, please correct it that you don't.
Yeah, sure, sir. Sure.
Second thing, I'm very curious to ask you about that in your title. Why have you selected antimicrobial tolerance? Why not resistance?
Because, sir, resistance is a complete, for resistance.
Okay, before you answer this question, you have taken 84 isolates, 84 or 85 isolates. You have categorized them into MDR, XDRs, etc.
Yes, sir.
Right? What was the criteria of this categorization?
Resistance to number of antibiotics.
How do you define MDR?
Less than 3 were considered as MDR, then greater than 3.
Less than 3 what?
Less than 3 different antibiotic group as such, resistance to antibiotic in 3 different groups.
Be very careful when you are answering. It's
Okay.
not the right thing.
Okay, sir.
When you categorize, when you define MDR, XDR, this is not the right answer.
Okay, sir. I'm not sure.
Anyway,
Okay, sir.
so you have categorized them into
Yes,
MDR?
yes.
Most of them were MDR?
Yes, sir.
Any strain which was XDR?
There was, sir. There were 2
There
strains. Yes,
There
sir.
was.
Yes, sir.
There was? Okay.
Yes,
Your
sir.
120 strain is MDR or is
120 strain is an XDR, sir.
it is an XDR?
Yes, sir.
So how many classes of antibiotics it is resistant?
Sir, we tested 14. After that, I think so, 13 or 12 it was resistance. Overall antibiotic, and the classes we tested, we total tested 14 antibiotics of 5 to 6 classes. So this 120 was resistance to, except for I think so, colistin, it was resistance to all antibiotic. And one more.
I am little apprehensive because in our work, colistin resistance is the most rampantly coming now. Colistin
Okay, sir.
resistant. And Cipro and all that, they are
Okay,
all
sir.
coming. When you said that you have taken 14 classes, they were exclusively for gram negative or they were for gram positive also you are including?
No, sir, for gram negative, I did for Pseudomonas during
I don't
the.
have that data actually, so I cannot comment much on that actually.
Okay.
Because if you say that, I take your word on that. Now I come back to my question again.
Okay.
Now when you are saying that you are working on the MDR and XDR strains,
Yes, sir.
why antimicrobial tolerance?
Sir,
Working on the resistant strains.
yes, sir. They are resistant, sir. But I wanted to know if tolerance, how the bacteria tolerate the antibiotic.
You know the difference between tolerance and resistance?
Yes, sir. If we give a sublethal concentration of antibiotic, then bacteria might survive, tolerate that amount of antibiotic, and it can still grow. So I wanted to understand if antibiotic concentration is decreased, if the strain gets a little bit of less amount of antibiotic, will be able to tolerate that antibiotic, and what will be its biofilm response during such antimicrobial tolerance?
Nobody is going to buy this argument.
Okay, sir.
It is you're working on the XDR strains,
Yes,
and you're
sir.
saying I'm working, the title is antibiotic tolerance. Nobody is going to buy your argument.
Okay, sir.
I will say that now it is okay. The title should have been antimicrobial resistance, and that would have been carrying more weight also, and that is correct also.
Okay, sir.
Right? That's
Yes.
my suggestion to you, and my
Okay.
experience telling you, because
Yes,
we
sir.
work
Sure,
a
sir.
lot on that actually.
Sure, sir.
Right? Every day we are finding, even in environment, in hospital settings,
sir.
in community also, we are finding Pseudomonas now.
Yes, sir.
Coming
Yes,
down. My
yes.
main work is on Acinetobacter baumannii, but
Okay, sir.
we work on acid. So take my word on that.
Sure, sir.
Right?
Okay, sir.
Okay. With this, let's move further.
Okay,
Your categorization definition of XDR, MDR, please correct it. This,
Okay, sir, I
needs
might
further,
have used correct, but I'm
maybe.
not remembering, and so I might not be presenting with you.
Anyway,
Correct.
I take it because at this moment many people are confused and nervous also. I take
so
that
I'm not
it's
remembering.
Okay, but that doesn't
Yes, sir.
undermines the work you have done actually.
Okay, sir.
It's a lot of work you have done on that actually. I'm not undermining that.
No, sir.
But
It's okay.
my job is to correct
Yes,
you
sir. Yes, sir.
wherever I can correct you. I
Yes,
tell
sir.
you whatever the right way is.
Yes, sir.
According to me. Throughout, you have done quite a bit of work on estimation of biofilm by CV method and
Yes, sir.
by.
Yes, sir.
I don't know why you have included risavurin and congruent. They are not good methods for biofilm estimation. Even CV. Let me first finish.
Okay, sir. Oh, sorry,
Then you
sir.
can
Sorry,
answer.
sir.
Even CV is not a good method for the biofilm estimation.
Okay, sir.
But because I can take that you have to do a lot number of samples, you have done CV. Otherwise, CV is a first step method.
Yes, sir.
Ultimately, we go for confocal only.
Okay, sir.
The confocals, they give you the best way how much your biofilm is formed.
Okay,
the
sir.
layers, the thickness, its whole matrix.
Yes, sir.
That
Yes, sir.
confocal is
Yes,
the
sir.
best way to go for that.
Yes, sir.
CV is a very error-prone method actually.
Yes, sir.
It's a very, very error-prone.
Yes, sir.
But still, you should have at least in some cases should have done confocal, so that you can validate whatever
Okay,
you are saying by CV
Yes,
is
sir.
directly coming on the confocal also, because nowadays good papers are not accepted on CV basis only, they take it the confocal.
Yes, sir.
Right?
Right,
That's
sir.
my
Yes, sir.
another suggestion. Then you had said that there were some strains which are out of 84, they don't produce biofilm.
Yes, sir. 4 strains.
Are there some strains which are not?
Sir, that only initial
It is
screening.
a little surprising that some strains are there which don't produce biofilm at all.
Sir.
It
Sir,
is
that
very
only.
surprising for me actually.
Sir, we used CV, so there was an error. We tested in different surface. In one surface, there were no biofilm. There were all biofilm forming strain. So it was with respect to the method we are testing and the surface we are testing. With that respect, initially we got 4.
That's I got it. Okay,
Yes, sir.
but then you have several things because you know that deeper you go, you have tried CV method with 3x washing, 6x washing,
Yes, sir.
and that also gives you a different result. Had
Yes,
it been
sir.
4x, 5x, all would have given you the
Yes, sir. Yes, sir. I understood.
very error prone. Very
Yes.
error.
Yes, sir.
You do with confocal, they will not give you
Yes,
such
sir.
error actually.
Yes.
So that puts a question mark.
Yes,
on
sir.
it.
Understood.
I would not have gone for 3x, 6x. That puts a question mark on your CV method actually.
Okay.
Right?
Yes, sir.
But you have tried it. It's
Yes.
a very good that you are true to your. Pseudomonas aeruginosa is known for alginate production.
Yes, sir.
You have shown me that video where when
Yes,
you
sir.
hit,
Yes, sir.
120.
Yes, sir.
So could you see that it's more producing more alginate, not the biofilm. It's just more producing more alginate.
Kind of,
and that's why.
we found that this must be alginate only because of the consistency which we are seeing. That difference should be alginate, as we found during the expressions also, that in PS120 ALGD was highly expressed, and thus that should be the reason. Alginate production should be one of the reason.
Okay. Then as you have done it yourself, I'm happy that you have given your limitations also at the end. That's nobody gives that actually, but you have given your limitations.
Yes,
That's
sir.
fine, because I was going to ask you, what is the fun of doing all these biofilm related studies in micro titer players? Ultimately, in the clinical samples,
Yes.
that doesn't go. It's not going to happen.
Yes, sir.
Catheters only okay, fine. But
Yes.
other lungs or tissues, fine. But micro titer players and
Yes,
all
sir.
that. Okay, this is just an academic, but you have yourself committed that, okay, this is
Yes, sir.
one of the limitations, not the biological samples you have done, so could have been done. Overall, I mean, I appreciate that you have done a lot of work on all these multi-omics actually,
Okay, sir.
but on the flip side, I also see that, don't you think that rather than expanding so much into this, could have taken a limited approach, but then finding or focusing more on the solution also. Your work
Yes.
is giving me the problems. These are the genes.
Yes,
this
sir.
is the problem. But where is the solution?
Yes, sir. Sir, I wanted to. My interest was in studying a basic biology. Wanted to understand.
No, I understand that.
So
but you understand that individually omics, several people have done.
Yes, sir.
You have combined
Yes, sir.
that people have done. But now people are looking for solutions.
Yes, sir. I
They
understand.
want to contain pseudomonas.
Yes, sir.
Right?
Yes, sir.
So that's
Yes, sir.
one approach
Yes.
where if I would have been your guide, I would have given, okay, let's go to the solution rather than finding more problems.
Yes, sir. Sir,
Anyway,
sir.
that is, again, I'm not undermining your
No, no,
study.
yes, sir.
It's absolutely fine. Very good what you have done.
Thank you, sir.
Not undermining, but again, a
Solutions.
suggestion
Yes, sir. I understand.
to that. Okay, then this, I was seeing your sequencing, because we do a sequencing every
Yes,
day
sir.
now,
Yes, sir.
every isolate. Your 120 strain is the largest genome.
Yes, sir. Yes, sir.
7.7 something.
7.2.
7.2 something.
Yes, sir.
Compared to 6 point something
6.
KPA1.
6.6.
Okay. Obviously, it has acquired so many HGT genes
Yes, sir.
or islands.
Yes, sir.
Did you find out which horizontal gene islands it has acquired?
Sir, in
Because
profiles.
in our first aim is, when I do all this study sequencing, which are the horizontally acquired genes or islands?
Okay, sir.
Did you find that?
Sir, that prophage we saw. That,
Prophage is okay. Prophage is generally, you will have found prophages were they carrying the antimicrobial resistance genes? No.
sir, in that it wasn't.
It wasn't. We know in our experience
Yes,
we
sir.
find lots of phages. but generally they are not carrying the genes.
Carrying the
for it.
genes. Yes, sir.
Did you find that antibiotic resistance genes which you are claiming are there in 120, whether they have been acquired or they were indigenous?
No, sir.
That would have been a better study.
Okay, sir.
Did you see our latest paper? If you see our latest paper
Okay,
on
sir.
Bacillus cerealis, where we have completely sequenced it and found that prophages were there, and
Yes, sir.
that we published in the International Journal of Abia. So
Yes, sir.
we focused on when the organism is obviously 7.7 means it has acquired lots of genes.
Yes, sir.
Lots of genes.
Yes, sir.
And when it is acquiring any organism, when it is acquiring a lots of genes, it is for its own benefit. It wants to evolve better against the antibiotic.
Yes, sir.
So could have done that
Yes, sir.
at GT genes, islands, pathogenicity islands, virulence genes,
Yes, sir.
where they were required. And then what we go one step further also, those genes which are required, then we do the evolutionary back study also, from which organisms, from where they have come.
Yes, sir.
Could have been added to your, it have made the study even better actually. That.
Sir, that one more limitation I have not added is that once we started doing this work, the combination we understood that there is lots of data.
Yes.
And so digging
But
into
that would
that
give
data.
you a better publication also, because
Yes.
right now I'm seeing that you have a very good review publication, but your publication
Yes,
of
sir.
this work is not that.
Not yes, sir.
This kind of work I'm telling you that would have given
Yes,
you better
sir.
publication.
Yes. Sir.
Okay. Then you have been talking about the motility also.
Yes, sir.
Motility genes, they go up also. Did you analyze that what kind of motility is going up in Pseudomonas? It has three types of motility:
Yes.
surface, swabbing, and twitching motility.
Yes, sir.
Did you delineate?
No, sir. We phenotypically didn't study.
Not phenotypically, but maybe the genes for it.
Sir, genes that type 4 pili results in twitching mortality, and we found the differences in mutations also, variations also between these two strains, and as well as expression changes also were different. So based on that, we understood that pili, type 4 pilus is one of the gene which is responsible for differences in finally we want to biofilm, whether it's forming high or low, or is it forming. So there was difference in this gene expression PILY1 between these two strains.
So the type 4 pili which are involved in the twitching motility,
Yes, sir.
was that high in 120?
Yes, sir. It was it was low in 120 as compared to PaO1, and in PS51 the twitching
But don't you
motility.
think that type 4 pili and twitching motility is more involved in virulence?
Sir, it is involved in virulence, but maybe because of the mutations, we found a lower expression.
These mutations which you are telling me, this is just a genomic study, actually
sir.
bioinformatics study. How do you know that these mutations are down mutations or up mutations?
Okay, sir.
You can't say that unless you validate them.
Yes, sir.
Any mutations which have taken place,
Yes,
they
sir.
may have,
Yes, sir.
given the
Yes, sir.
more activity. If
Yes, sir.
expression is one thing, activity is another.
Activities, yes, sir.
Right?
Yes, sir. Understood, sir.
Okay.
So, that we not.
You said that ciprofloxacin exposure,
Yes,
it
sir.
has caused the cell wall modulation.
Yes, sir.
I could not understand the correlation, because ciprofloxacin, you know that it's not a cell wall inhibiting
Yes, sir. Yes, sir. Yes, sir.
antibiotic.
Yes, sir.
It works on gyrases,
Gyrases.
right? You know that.
Yes, sir.
But why did you think that there should have been a cell wall modulation?
Sir, there was cell wall porine, so it doesn't allow the ciprofloxacin to go in initially at first stage only because of the modification. It doesn't allow only the ciprofloxacin to go inside only. Then only because of that modulation, and then if it goes inside, then other genes might be activated.
But
so one
in
of
my
the
opinion, cell wall modulation is a different thing from the efflux pumps.
okay, sir.
What you are saying is the job of the efflux pumps.
Okay, sir.
Cell wall modulation is different. Like in the presence of cholestins,
Okay, sir.
they may happen, but here I don't say they could have happened, but I'm just trying to understand that what could be the reason.
One of the
Because when
reasons.
efflux pumps are there,
Okay, sir. Efflux pumps it's outside.
basically majorly it is the efflux pumps.
Yes, sir. It pumps outside.
Oh.
Yes, sir.
Actually, okay. One more thinking. Throughout, you have been watching it that whichever genes you are taking, biofilms, formations, or whatever, in 120 consistently, all the genes are upregulated.
Yes, sir. Many of the for the limited RNA expression studied we studied, many of them were highly as compared to PS51.
What could be the reason that ki sari genes they are upregulated in 120 and not in 151?
Sir, हाँ वही बहुत सारी थी, especially related to quorum sensing and matrix related. Sir, that only we are still deciphering using that we studied. We wanted to study all these integrations together to find a way that actually how it works, so that integrating bioinformatics is still remaining. Conceptually, I guess it as found in proteomics, the PS51 goes for conservative nature. It conserves the protein expression, and it while PS120 goes for alternate metabolism. It uses alternate metabolism, and some it is kind of what you say head-on taking and fighting, while PS51 is conserving, kind of suppressing when it encounters ciprofloxacin.
No, I think this may not be. This is more of a conjunction actually.
Okay, sir.
I don't see it because possibly because your genome size is so much, so many genes would have been acquired, and from where they have been acquired, they might be having stronger promoters, maybe expressing more, maybe.
Okay, sir. Yeah.
Couldn't it be one reason?
Yes, sir.
Otherwise, all the genes are getting activated in higher level. That makes, had it been the inherent genes, that makes a little surprise.
Okay, sir. Promoters, certain promoters.
Maybe.
Yes. Okay, sir.
I mean, in that case, you can not here, but you can in some later studies you can study the promoters, minus 10, minus 35, and the upstream sequences, and you can find out why they are hyperexpressed.
Yes, sir.
You can study that actually. Chalo, this is quite done. Finally, now in this age of artificial intelligence, we are also, so we have done lots of proteomics and all these things, kinds of genomics, transcriptomics. Finally, what is your view that now in this age of AI, when AI is everywhere, we are also in microbiology now putting AI in finding vaccines, in finding new antimicrobial peptides. For example, I tell you, we are working on antimicrobial peptides against Pseudomonas and Acinetobacter and all that.
Sir. Yes, sir.
And we design them through artificial intelligence.
Yes, sir.
And they work very well in vivo. Do you think that looking at all this data, AI needs data.
Yes, sir.
You have all this data.
Yes, sir.
Now in future, apply AI and try to find out the best of the targets.
Yes, sir.
Can you do that?
Yes, sir. Sure. I am interested in AI, and that's the challenge that.
And those best targets, then you go for the in vivo study. Otherwise, you have lots of targets.
Yes, sir.
You have given us lots of targets. But, just
which would be the
best ones?
Best ones. Yes, sir. Sure, sir. I can do it. Okay. We are planning to combine all this data and analyze how it can be best used. Sir was telling that next PhD we had focus on that only. That once we do this study, next PhD student will focus on the target. So we are planning on.
I tell because I teach BSc first year also, I teach MSc, all the classes. When students nowadays come to BSc first year, I tell them there was a time when computers came and people used to say that कि जिसको कंप्यूटर नहीं आता, he is illiterate. Now I tell them जिसको AI नहीं आता, he is illiterate. Whether it's microbiology or scar making or wherever, if you don't know AI, you will be nowhere.
Yes, sir.
So in microbiology in our university now we have started AI courses also in syllabi or AI based research also. Otherwise you will be nowhere. That's why good work, good data. Apply AI on that. Find that best of the targets. Try in vivo studies.
Sure, sir. Thank you, sir.
Good. That's all I wanted to talk about this. Good.
Thank you, sir. So much appreciated. You came and gave.
No, that was my job actually.
But I appreciate, sir, that you give.
I mean, yes, there can be lots and lots of discussions because मेरा field ही है AMR पे.
Yes, sir.
But I think let's stop here now. And I congratulate you. You have done a good work. Please.
Yes, sir.
Okay, so this is not my area. I'm a biochemist toxicologist.
Okay, sir.
Just few friendly suggestions.
Okay, sir.
So number one, when an expert, I know this quorum is the most important one for you today.
Okay.
But whenever you are in public speaking, you should listen first.
Okay, sorry.
When the expert is talking, you should not interrupt. Listen to the complete question.
Okay, sir.
Right? Absolutely. That's
Sure. Sure.
Number two, when you're speaking in a public forum, doesn't matter if the room is occupied by 4 people or 3 people, right? So you should address everybody in the room.
Okay, sir. Okay,
So
sir.
everybody was there. You focus as I understand, you focus on mostly on the 4 people.
Yes.
Rather like
Yes, sir.
Just
Sure.
friendly.
Thank you, sir.
I have 2 questions here.
Sure.
So, and as I said, you excuse my ignorance on this. So I've seen a couple of points here about the redox sensitive pathways of oxidative stress.
Yes, sir.
I just need to, this might be a very stupid question, but I just want to ask, if oxidative stress kills bacteria,
Yes, sir.
then why do biofilms still survive? Why do biofilms still survive?
Because the pathway, to counter the pathway, various enzymes are activated, catalase and that, which could deregulate this pathway, and the one which is capable of in stress working and regulating the pathway which could deoxidize this has a greater chance of forming biofilm and survival as compared to the one which cannot form biofilm or cannot.
It might be because biofilms might have different multiple protective systems, right? Besides oxidative stress is just
It's one of the way.
one.
Plus mechanical barriers are there and various others protective mechanisms are there
Correct.
with respect to oxidative stress.
I've seen one of your slides, objective 1.
Okay, sir.
I've seen it in your presentation. So it's just a very basic question. So how would you distinguish whether a biofilm initiation is triggered by mechanical surface sensing or by secondary changes like intracellular signaling? Like you mentioned calcium GMP. So
Yes, sir.
how do you distinguish between those two?
Sir, the changes starts when the pili and flagella and all, they find the surfaces, and once the surface is there, then the VFR pathway, which is the one which is regulating the surface sensing, that triggers an activation signal, and that then triggers the initiation then phase. So that sensing is the first part. After that, there is a changes in such various regulatory pathways, which then goes into forming biofilm as such.
Usually, you want to ask a question. Already addressed, I think sir already addressed. I think.
Who has to do it?
Conclude.
Yes, sir.
Okay, to conclude this, I will say that Mustafa, you have done a very good work.
Thank you,
A lot
sir.
of work has been done, and with the multi-omics approach to understand completely, comprehensively the biofilm formation, which will ultimately result in the finding the newer targets
Yes.
for therapeutics and more importantly for diagnostics. Also in case of Pseudomonas, which is as you know among the WHO's priority list
Right.
of pathogens as far as its virulence and its hospital-acquired infections are concerned. So I will say that try to find good publications out
Yes, sir.
of that. You
Yes,
have
sir.
done some already into this, and with this type of a good work and something about the therapeutics, I think
Yes,
should be coming out.
Yes, sir.
And more of the in vivo studies in future will be coming out of this. So I congratulate you that you have done such a good work, and
Thank you, sir.
I say that you should be now offered a PhD degree into this, and you should be called now Dr. Mustafa.
Thank you, sir. Thank you. That means a lot. And thank you for guides also. They helped me. They were motivated for me to perform.
Of course, it's always there with the guide.